Awesome. It's my great pleasure to introduce Professor Tara Spires-Jones. She is the director of the Center for Discovery Brain Sciences at the University of Edinburgh and the recent president, I think, until as of last summer, if I remember correctly, recent president of the British Neuroscience Association. So Professor Spires-Jones has dual bachelor degrees in biochemistry and French from UT Austin. She then completed her master's and PhD at Oxford, where her dissertation advisor was Sir Colin Blakemore. She then moved to MGH, a place I'm familiar with as well, for postdoctoral work with Bradley Hyman, where she stayed on as an instructor and then assistant professor in her independent position there. In 2013, she moved to the University of Edinburgh, which really I consider to be probably the best city on earth. Um, and in Edinburgh, her lab's research focuses on the mechanisms of synapse degeneration in Alzheimer's disease, as well as other neurodegenerative diseases. Her group has done some phenomenal work. They've shown how amyloid beta and tau contribute to synapse loss, and especially how pathological forms of tau can synaptically spread through the brain. In addition to all of her phenomenal research, Professor Spires-Jones also does a brilliant job communicating new findings to the public, um, and is often called upon by major news outlets, including the BBC, where I've seen her quite a few times being interviewed, to really distill the latest potential treatments in ways that are easily interpretable to broad audiences. So delighted to have her here, and without further ado, here's Professor Spires-Jones to tell us all about synaptic tau.
Thank you very much, Omar. It's really nice to see you all, and thanks for the invitation. Some of these are brand new data, so if it's not the smoothest presentation, I apologize in advance. And I agree with you, Edinburgh is one of the greatest cities on earth, but it's very dark, so apologies. I'm at home in this home office, and the lighting's not very good, but you can imagine that I'm in a better lit place. So these are my declarations of interest, and it's an opportunity to thank the funders, the lab, and especially the brain tissue donors. Almost all of the data I'll show you today is from human postmortem tissue or human tissue from surgical resection. So what I'll talk about today is a little bit of an introduction, and then I'll talk about transsynaptic spread of tau and Alzheimer's disease, PSP, and then I thought for your audience, I thought there might be a couple of people interested in alpha-synucleinopathies in your institute. So I thought I'd put in some of our new data that's not published yet on how we see tau and alpha-synuclein in some of the same synapses in synucleinopathies, and they might even be interacting. So I'll get to that at the end. And this is just an excuse. Oh, no, our pointer's gone to show you one of my favorite images from Caitlin Davies when she was a PhD student. She just took some amazing images of synapses, which are in white and green, and this is a tangle in red, and there's dappy in blue. So, but bear with me for a minute, because as Omar says, I love a bit of public engagement, and this is sort of where I start when I'm giving public engagement talks. I like to talk about the history of dementia research. In fact, I like it so much that I put a lecture on it in my elective course, and I've written a book that I'll come to at the end, and I have a chapter on this in this book that I've written. But the reason I like this so much is because usually when you hear a talk about dementia research, people think about 1907 and they think about Alois Alzheimer, right? But I want to convince you in the next 2 minutes that the history of dementia research goes back thousands of years, and it's a really multicultural history. And I think that's important to acknowledge. And you have to sort of give me a little bit of leeway because I'm not talking about in 2000 BC fundamental neuroscience research, but I think we could argue that before we can discuss a disease or understand a disease in terms of modern research, we need to at least define it and know the symptoms. And the first place I could see mention of the symptoms of dementias when I was going back through a whole bunch of literature on this was in ancient Egypt. So the ancient Egyptians had, I have a quote here, sorry, I've got 3 screens. They described symptoms in old age as a perishing of the mind and forgetfulness, which I think is still pretty apt today. This is not a symbol for dementia, that's actually the symbol for brain hieroglyph, but it's very cool. And then if you look back through Indian traditional medicine, there are texts that describe not only the symptoms of dementia, so memory loss, etc., in aging, but also of herbal treatments trying to help people living with these symptoms. The ancient Greeks, of course, got in on this. So Plato talked about dementia symptoms, and he talked about in old age that old age can give rise to all manners of forgetfulness as well as stupidity, which is maybe not very kind, but could be considered accurate. The ancient Chinese text, this is the Yellow Emperor's Internal Classic, also mentioned dementia symptoms. Hippocrates, so still the ancient Greeks, came and talked about these symptoms as well, but also Hippocrates and Galen talked about them maybe having something to do with the head. So they were even starting to think about where physically these symptoms might be originating. And then during the Islamic Golden Age, Avicenna wrote about dementia symptoms. And then we start to get into the modern era where we start thinking about dementia as we describe it today. So Pinel and Esquirol were psychiatrists in France and they defined the symptoms. So you're probably all working in something to do with neurodegenerative research, I'm guessing. And that means you'll be, even if you weren't, you probably would know the symptoms of most dementias are this progressive cognitive decline. You start at a normal cognitive baseline and you steadily lose function. And with that framework of being able to diagnose formally, then fairly quickly, scientists moved on to being able to look in the brain. So in the late 1800s, there were advances in microscopy, compound microscopes were invented, formaldehyde fixation was invented, the silver stains that are still used today in many neuropath labs that can label things in the brain were invented. And that's what allowed people like Alois Alzheimer, Solomon Carter Fuller, and Oscar Fischer to actually look in the brains of people who had dementia. So the disease is named after Alzheimer, obviously the most common form of dementia, but it could have easily been named after Oscar Fischer, who was the first person to really describe plaques in detail. He was a student of Pick's in Prague, whereas Alzheimer was a student of Krapelin's in Munich, and they had this big sort of feud. And Fischer was killed by the Nazis, actually, so there was not a huge chance for his career to flourish. And I like to point out Solomon Carter Fuller. He was an American born in Nigeria. So he was the first Black psychiatrist, I think, in the US, and he trained under Alzheimer. And Alzheimer first described Alzheimer's disease in one person, August Dieter. But Solomon Carter Fuller came back to Boston. He took a ton of autopsy cases and turned that case study into a really well substantiated pathological entity. And in fact, he first described plaques in elderly people without dementia symptoms. He described glia around plaques, so another really important figure. And since then, there's been an explosion. There's, can't go through everything in dementia research. In the 1980s, is when the molecular revolution was happening and we started to be able to get at the proteins that are in the pathological lesions that I'll come to. But it really took until 2023 and 2024 for the first disease-modifying symptoms for any dementia to be, disease-modifying treatment rather, for any dementia to be approved. And those are lecanemab and donanemab, and they're not great, but they do slow disease progression by removing one of the pathologies that was described by these three people in the early 1900s. So that's the history of dementia research. What is dementia? It's the umbrella term for the symptoms that we're all familiar with. And dementia is caused by lots of underlying pathologies. Alzheimer's is the most common, and we'll talk about that today. But we'll also talk about progressive supranuclear palsy, which is a type of frontotemporal dementia with some motor symptoms. We'll talk about Lewy body dementia as well. And one of the reasons that this is an important problem is not just because it's fascinating and important and the symptoms are very, very difficult, but because as our population ages, with age being the most important risk factor, the prevalence of the disease is growing dramatically. And it really does have the potential to bankrupt our healthcare systems if we can't get a handle on it. These are some data from the Global Burden of Disease study in 2021, and I just plotted them out in R. So the prevalence, more red is higher. And you can see in first world countries with the aging populations is where we have the most dementia cases. Japan has got really high incidence. But even in the low and middle income countries, our populations are starting to age, and they are less well equipped in many ways to deal with the sort of people living with dementia. So it's a problem for all of us. So we'll talk about Alzheimer's disease first. And in case you are not aware, these are those pathologies that Alzheimer and Fisher and Solomon Carter Fuller, sorry, it's late here. I can't get names out, first described. So there's still three definitive neuropathological lesions we use to define Alzheimer's today. Those are atrophy, which you can see on the top. So that's just a coronal section through a postmortem brain showing you that the person with Alzheimer's disease had massive shrinkage. So that's neuron loss, synapse loss, white matter degeneration. And the two microscopic lesions that Alzheimer and Carter and Fisher described were plaques and tangles. So plaques, we know since the 1980s and 90s, are made of sticky amyloid beta peptide that accumulates extracellularly, and tangles are made of hyperphosphorylated tau protein that accumulates inside cells. But these don't show up all at once. As anyone who's met someone living with Alzheimer's knows, they're very slowly progressing diseases. And what we can see in the brain, at the y-axis, I'm going to show you the amount of stuff in the brain, and the x-axis is time. Prodromal is when you have changes in the brain but no symptoms. MCI is mild cognitive impairment, where you do have symptoms. but they're not affecting your activities of daily living. And then the gray on the end is when you have an Alzheimer's diagnosis. So age is the most important risk factor for Alzheimer's, and all of us as we age, or most of us, will have gliosis. So we'll have changes in the glia in the brain. And this ramps up a little bit after pathology starts in Alzheimer's. Amyloid beta, these plaques that you can see on the left here, ramp up really steeply in the prodromal phase. And they actually get to a plateau at about the time that symptom onset can be detected with neuropsychological tests. So for about at least 10, probably more like 20 years or more, amyloid's building up in the brain before you have any symptoms. Tau pathology, there's a little bit in the medial temporal lobe in most of us as we age, but really when symptoms start at MCI, that's when the tau pathology really takes off in the brain, and it tracks much more closely with symptoms than amyloid or gliosis. Synapse numbers start to go down around plaques as soon as plaques appear. That's something that I'm not going to talk about today, but we've done a lot of work on plaque-associated synapse loss. Actually, we might. I'm not sure if I put in. Sorry, it is late. We're focusing on tau. That's right. And neuron numbers start to go down as tau progresses through the brain. So where tau goes in the brain, the neurons die. So this is a very slowly progressing disease. After a study about Alzheimer's, I'll tell you a little bit about what we've done in PSP. So I'll introduce progressive supranuclear palsy. If I were betting money, there are 20 odd people in this Zoom room, and I would bet that most of you have met someone with Alzheimer's and fewer have met someone living with PSP. It's a less common disease. It's characterized by impairments in movement, balance, as well as cognition and behavior, the sort of dementia-like symptoms that I've talked about. The prevalence is only between 1 in 18 and 100,000, so it's much rarer than Alzheimer's disease, but it shares one core pathological feature, which is the accumulation of tau pathology. In PSP, we do see neurofibrillary tangles like you do in Alzheimer's disease, but there's also tau pathology in oligodendrocytes in what are called coiled bodies, and there's tau pathology in astrocytes. I actually see tau pathology in astrocytes in many diseases. We see those in Alzheimer's as well, but they're definitive for PSP and other primary tauopathies. And finally, dementia with Lewy bodies. I'll talk a little bit about dementia with Lewy bodies and Parkinson's disease at the end. DLB is another type of dementia. It's characterized by not only the cognitive decline that we're used to seeing in people with Alzheimer's, but also visual hallucinations, sleep disturbances, and Parkinsonian symptoms because it overlaps on a spectrum with Parkinson's disease. They're both alpha-synucleinopathies. Alpha-synuclein accumulates in Lewy bodies, but also in Lewy neurites in these diseases. So that's the three diseases. And what I'm going to talk about is synapses. And this is a block of cortex from a human brain that's 30 by 30 by 30 microns. And a really talented EM technician, John Moss, was able to take a tiny, tiny block of brain and do 3D scanning block face electron microscopy. And I'm showing you this because you can see how packed the brain is, right? You can see axons running through, you can see myelination, you can see there's not a single cell body in this image. And so this is neuropil. And on the right, I'm showing you a single synapse. So you've got 100 trillion or so synapses in your brain. And of all the pathological stuff that I just told you about, it's the loss of these synaptic connections that most strongly tracks with dementia symptoms. So this is what we're focusing on in the lab. One of the reasons is because, as you know, as neuroscientists, synapses are also very plastic. We can make new connections in the brain, we can make new synapses, whereas once we've lost the neurons, it's much harder to get neurons back into the correct circuit. So we think they're also a really promising therapeutic target. And here's a little bit of the evidence that suggests they're important. This is in Alzheimer's, PSP, and DLB, the three diseases we'll focus on today. And then showing you essentially the same thing in different kinds of graphs. So for Alzheimer's disease, this is one of the original studies looking at synapse density using EM, like I showed you before, by Steve Sheff and Steve Dukowski. And what they showed, unsurprisingly, is that the more synapses you have, the better your exam score is on the MMSE, the mini mental state exam. So more synapses is a good thing. This is showing you change in a different cognitive test called ACER, correlating with UCBJ, which is a cool, fancy way of measuring synapses in living people. So it's a PET ligand that binds presynaptic vesicle protein, SV2A. And this was Mauro Malpetti and James Rowe's work showing longitudinal synapse loss correlates with cognitive and motor symptoms in PSP. And similarly, in dementia with Lewy bodies, both subcortical and cortical synapse loss measured with SV2A PET correlates with your symptoms. So it's a very important feature correlating with your symptom progression. Another idea I want to stick in there before we jump into tau is this idea that it's not just neurons in the brain. When I started in the field 20 odd years ago, we were all focused on neurons, and I was focused, of course, on synapses. But what we've come to appreciate from the genetic data is that glia are probably driving at least the risk for these diseases. So this is the Alzheimer's disease genetic data. And what I've done here is shown you all of the gene, well, no, not all, a few of the gene variants associated with risk of Alzheimer's disease. These three all influence a beta accumulation and they cause familial Alzheimer's disease. But the variants that I've colored in pink here are really predominantly expressed in non-neuronal cells, either microglia, astrocytes, oligodendrocytes, OPCs, or in rare cases, vascular cells. And you can see there's just a field of pink here. So most of the genes that influence your risk for developing Alzheimer's are not even expressed in the neurons. They're expressed in the glia. So one of the things I'll talk about is synapse-glia interactions, and a big part of my lab is focused on that. Oh, and I've got a little star here because I'm going to show you a little bit of data on BIN1. This is one of the risk gene variants that increases your risk for Alzheimer's disease, and we think it might have something to do with tau and synaptic tau. And now to tau, just to give you a brief introduction to my favorite protein. Tau is encoded by the MAPT gene, and I've made a cartoon of it here in a nice branded Dementia Research Institute colors, so I think I should get extra points for that from my grant funding body. So I'm showing you here that the constitutive coding exons are in blue. There are 3 alternatively spliced exons in the brain that I've colored in pink here. And what I'm showing you, all these tiny little notations along the side, are all variants that can cause primary tauopathies. So all of these mutations can cause neurodegenerative diseases influencing tau pathology. And the translation of the tau RNA makes 6 different isoforms of tau protein in the brain based on the inclusion or exclusion of those 3 alternatively spliced exons. So the longest form has both exon 2 and 3 and 10, et cetera, et cetera. So we have these 6 isoforms of tau. And the longer tau forms with the more microtubule binding repeats are generally stickier. So they're more prone to aggregation. But there are diseases that have either 3 repeat or 4 repeat tau inclusions. Alzheimer's disease has both, has 3 and 4R tau in the tangles. And when tau is first translated, it's made into 1 copy, obviously, of tau. In the disease process, tau becomes aberrantly overphosphorylated. That causes it, we think, to fall off the microtubules, stick to itself and form oligomers, and eventually this forms filaments that make those intracellular tangles that were described over 100 years ago. Turns out that the big lesions that people have been measuring in the brain and using to stage the disease for a century are probably not the thing that's killing the cells. When tau is present in a brain region, neurons are dying, but it's not necessarily the cells with the tangles that die. It turns out from work from lots of people in the field, including Christian Lasagna Reeves and Raquez Kayed, that it's probably soluble form, some oligomeric form that's actually bioactive and toxic to neurons. So we look at oligomeric tau in some of the studies I'll tell you about today. And this is, again, an excuse. I do love electron microscopy. So whenever I'm looking for synapses on the EM, if I see a tangle, I have to have a picture. So these are paired helical filaments in a human brain. So you can hopefully see, this is the cartoon version, looks like a bit like DNA, these paired helical filaments, and you can see them running through this tangle. That's a mitochondrion for scale, 200 nanometers. So the final thing about tau that I want you to know is that it spreads through the brain, and wherever tau goes, cells are dying. So in Alzheimer's disease, we start in the brainstem and the transentorhinal cortex in very early stages. Most people who are elderly will have this kind of pathology, and it doesn't really affect your cognition unless you also have amyloid pathology, and then the tau spreads through the brain. And when the tau spreads out through these stages that were described by Brock and Brock, the symptoms get worse, and as the tau progresses, the neurons die. PSP also has tau spreading through the brain. It's a little more complicated. PSP is a more complex disease in terms of the clinical presentation. You can have several different subtypes, but in general, the pattern seems to be, this is data from Gabor Kovacs, in general, you seem to go from midbrain and brainstem upwards and outwards, the tau pathology in PSP. And in both Alzheimer's and PSP, Nico Franzmeier and colleagues have shown that if you use resting state functional imaging, that combined with tau PET, you can see that the tau pathology is spreading through brain regions that are functionally connected. So this is more of an argument that we've shown in mice, et cetera, that tau can spread through synapses, or at least through circuits that work together. And I won't go into our mouse data, but we and many other groups around the world have shown, I think, entirely conclusively that tau can move from one part of the brain to another by jumping out of one neuron through the presynapse into the postsynapse of another neuron. And I'm showing you an example here where we've injected tau in the entorhinal cortex of a mouse, and the tau is, the virus has a GFP in it, and then we stain for human tau with a red fluorophore. And we inject it here, but you can see that where the axons project, you get tau down here in the hippocampus, and here in the dentate gyrus, particularly, which is the first relay in mice between entorhinal cortex and hippocampus, you start to see neurons that are positive for tau that aren't positive for the virus. So this is evidence in animals that tau spreads transsynaptically. But the question we wanted to get at is, was this happening in people? Because in all of the mouse lines that we used, and most of the ones that have been used at all for this kind of experiments, you're overexpressing the human tau. So that's a big caveat. You can imagine if you're causing a neuron to make a huge amount of a protein, it might spit it out. So that we might be driving this transsynaptic spread artificially. So we wanted to get some hints from human brain about whether this mechanism is active in human cortex or human brain as well. So what I'll talk about, that was, apologies, quite a long introduction, but what I'll talk about is these three hypotheses that tau accumulates in synapses in these diseases that we talked about, AD, PSP, DLB, and PD. For AD and PSP, we think tau is spreading in the human brain, at least in part, through synaptic connections. And I'll give you a little bit of data about how we think astrocytes might be contributing to this. So how do we do this? This is a video from our lab. One of the things that's troublesome about studying synaptic proteins in humans is that synapses are really, really tiny. And I showed you some EM, which is the gold standard way of doing this. But with standard EM, you can't stain for proteins, and it's very, very difficult and low throughput. So what we did is we worked with Steve Smith and Christina Makeva to incorporate into the brain banking process for human tissue a technique that they called array tomography. So this is me cutting up a tiny bit of cortex, and you can hopefully, if you have a good enough screen, see that there's a bit of white matter. We go all the way to the pia. So we're just taking very small sections of brain, and we fix them briefly. We have to take the tissue at autopsy. We fix them briefly, dehydrate them, and embed them in an EM-like resin called LR white. And this allows for immunostaining. But the reason we do this is because when we take that resin and bake it, it turns into a hard plastic that you're seeing there. And the reason we go to all this trouble, it's 3 days of processing every time we get a brain donation. And you can imagine it always happens on a Friday. So we're always doing this over the weekend. But once we get this tissue into this hard resin block, we can use this diamond knife here to cut sections that are smaller than an individual synapse. And XY plane of light microscopy, synapses are okay. You can see them, you can define them. But in the Z direction, even really fancy microscopes can't resolve individual synapses. So here what we've done is physically sectioned through the brain so that we have a section that's smaller than an individual synapse. And then we can just do standard immunofluorescence. So this is just putting on antibodies and rinsing and blocking and things like that. And then we could take it onto a microscope and we can look within individual synapses. So we can answer that question, is tau inside synapses? And here's sort of the experimental design for the first study. So in the first study, we looked at 2 brain regions, the medial temporal lobe here, this is BA2021, inferior temporal gyrus, which should have pathology fairly early on in the disease, about Brock stage 3. And we also looked at primary visual cortex way back in the occipital lobe, which gets tau pathology at the very, very, very end stage of disease. So we would be maybe thinking at end stage, we might still be capturing some spread. So from each person, we had about 20 people in the study, and we looked at 2 brain regions. And from each brain region, we looked at 2 of those blocks that you saw. From each of those blocks, we looked at 3 different image sort of stacks. And that meant we had over 1 million individual synapses in the experiment. But because we wanted to make sure that we are statistically rigorous, we use these linear mixed effects models because we want to use all those million synapses, but we want to tell the stats model they came from each block or each person. So this is how we run our stats. And here's the paper that this first bit of data was published in. It was published 2-3 years ago now. And we're all very happy because the Alzheimer's Association awarded the paper a prize. So we had to celebrate. These are the co-first authors, Marty and Caitlin, and myself and Alberto Leo, the co-senior authors of the paper. So I won't go through the whole thing because it was published a few years ago, but I can show you that a few of the key experiments. So we looked at oligomeric tau using an antibody from Raquez Kayed, and whether it was present in synapses. And in this first experiment, we were actually head-to-head comparing oligomeric tau phosphorylated at a couple of sites. This 88 antibody is used to Brock stage pathology cases, and a misfolded form of tau with an antibody from Peter Davies. And when we quantified this, what Elizabeth and Marty saw was that about 4% of post-synapses contained oligomeric tau, and that was significantly higher than in the control cases. This was only an N of 9 per group in this part of the experiment. Phospho-tau and misfolded tau were also present, but to a lesser extent. So we followed up with oligomeric tau because it was most common and because of the data in the field suggesting it's probably the most toxic form of tau. We had some reviewers who wanted us to validate our array tomography technique. So we did that again, going back to immunoelectron microscopy. Jamie Rose did all the embedding, and Yayan Chang did a heroic amount of electron microscopy imaging. So with this, we've got an antibody to oligomeric tau, and then we've got a secondary antibody that has a gold particle on it, a 5 nanometer gold particle. So where you see these little clusters of black dots, that's tau staining. And this is inside a postsynapse, that's the postsynaptic density. The pink is inside a presynapse, so this is along synaptic vesicles. And then, of course, we see oligomeric tau also associated with fibrils in tangles, etc. When we went back to the array tomography and then quantified whether tau was in presynapses, postsynapses, or both sides of the synapse, this is the sort of indirect measure that we were using to try and assess whether tau could be spreading transsynaptically. So we were looking in this condition where we have presynapses are magenta, post or cyan, and tau is yellow. So in this condition, you have tau associated with the presynapse. This synapse had tau on both sides, and this synapse had tau only on the postsynaptic side. So we were defining synapses by proximity of a pre and postsynaptic terminal in this case. And what we observed is a little bit busy, but in both brain regions, that's temporal and occipital, and in both control cases and people with Alzheimer's disease, when tau was present at a synapse, which remember was only 4% of the time, but this 100% is all of those synapses that contain tau, it was most often only in the presynaptic terminal, and then there was a lesser proportion that had tau on both sides of the synapse, and even less with tau only on the postsynaptic side. This is, of course, a snapshot at the end stage of disease, so we cannot prove that this tau was traveling through the synapse, right? This could be coincidental tau arriving in the pre and postsynaptic side from each neuron individually. But if we never saw these synaptic pairs that had tau on both sides, it would be hard for us to argue that tau was spreading transsynaptically. So this is some indirect evidence that tau might be spreading through synapses in human brain from postmortem tissue. We also saw this out in the BA17 region where there were practically no tangles, and in fact, in some cases, there were no tangles, but we were already seeing presynaptic tau and some synapses that contain tau on both sides of the synapse. So this is some indirect evidence or circumstantial evidence that tau could be spreading through the brain in Alzheimer's disease. And then we went on to look in PSP, which is progressive supranuclear palsy, and this was published just last year now. Rob McGeechan was the PhD student, a clinical PhD student who led the study, and Dr. Claire Durrant was my partner in crime. She's the co-senior author, and we'll come to some of her very cool experiments at the end. So again, this is the same question, different disease. Could tau be spreading through the brain in PSP by jumping from presynapses to postsynapses? So we used almost exactly the same methods. This is now, instead of Alzheimer's cases, it's PSP cases. PSP is quite rare, so only had 7 people who we had tissue processed for this technique. But we stained again presynapses, postsynapses, tau, and astrocytes in this study. And what I'm showing you here are the T22 to the oligomeric tau data. And again, just like in Alzheimer's, we have some synapses that have presynaptic tau, some synapses that have postsynaptic tau, and some synapses that have tau on both sides of the synapse. And quantifying this is now showing you the proportion of synapses that have oligomeric tau in the presynaptic side and the proportion in the postsynaptic side. It's lower than we saw in Alzheimer's disease, so we see less synaptic tau, but it is still significantly more in PSP than in control. In this study, it was actually quite interesting. Rob showed that there's a correlation between synaptic tau accumulation and synapse density. So the more tau you have, the less synapses you have, so indicating that this synaptic tau is quite toxic, that we would have expected. Lois Keavy, during her master's, had a look at whether any of these synapses were getting eaten by astrocytes. I didn't go into the Alzheimer's study on microglia and astrocytes for the sake of time, but no one had really looked in PSP before at synaptic engulfment by astrocytes. And what Lois and Rob found was that the proportion of synapses that were co-localized with astrocyte processes went up in PSP. We validated with EM that at least some of these astrocytes had fully engulfed the synapse. So it wasn't just the astrocyte was contacting the synapse to support it, but as you can see here, this is an astrocyte nucleus. This is the lysosome of an astrocyte. You can appreciate from this low mag image that human postmortem tissue is not the best quality for electron microscopy, which is why it works better for us to use the proteins, which are better preserved. But we still can find these situations. So here I've zoomed in on the lysosome, and in this box I've zoomed in even further. Right next to the lysosome, now what we've got is labeling for synaptic protein, synaptophysin. So all these gold dots are synaptophysin. This little circle here looks very much like a presynaptic terminal and has little vesicles in it labeled with synaptophysin. And that is right smack in the middle of an astrocyte cell body. So these astrocytes are engulfing, phagocytosing, and eating synapses in the human PSP brain. And when Rob wanted to know whether tau could be spreading, back to the core question, He was looking at the proportion of PSDs that co-localize with T22, and he had a really clever way of thinking about this. So instead of just looking at pairs of synapses, he said, if we look at the proportion of all PSDs that have tau, it's less than 1%. I've shown you those data already. But if we look at the proportion of PSDs that have tau when the presynaptic partner has tau, it goes up by 84-fold. So if you have tau in the presynapse, you are 84 times more likely to have it in the postsynapse. So that's, to me, again, it's still postmortem, so it's still a snapshot, but some pretty convincing evidence that tau is likely spreading through synapses in the brain. And what made it even cooler in this part of the study was Claire Durrant's lab. So Claire works with Paul Brennan, who's a neurosurgeon in our unit, and Paul takes tumors out of people's brains every Monday. And usually what he would do, he has to go through part of the healthy cortex to get to tumors that are subcortical. And usually he throws away that access cortex, so it goes straight into the bin as waste product of the surgery. But because Claire was able to get a good amount of funding and a research nurse and ethical approval, what we're able to do if the patients consent is Claire or Rob or one of her team goes and stands in the surgery behind Paul. And when he would normally drop that bit of cortex that doesn't have a tumor in it in the bin, instead they drop it into a dish and they run it back to the lab. They slice it, and Claire's team can keep these human ex vivo brain slices alive for a few weeks. So they can do electrophysiology, they can do all sorts of stuff. So we've got living human brain, which is pretty amazing, I think. And what we did in this experiment is Rob challenged these slices of neocortex from a living human with either nothing, just the media to keep them alive and more or less healthy, or with postmortem PSP brain that he'd squished up and either immunodepleted the tau from or left the tau in, so mock immunodepleted. So what we're doing is we're challenging living human brain slices with disease-relevant tau. And the question was, can living human postsynapses take up tau? So this is the idea, instead of it being coming out of the presynapse, because this is not someone who had PSP, it's we're providing that toxic tau and seeing if it can enter the postsynapse as part of that would have to be able to happen if our hypothesis is correct. So what he did was he treated with those tau or immunodepleted tau conditions for 3 days, fixed the slices, and then again, we're doing the same array tomography experiment in the fixed slices. And so we're just staining for postsynapses, oligomeric tau, and phospho-tau, as well as astrocytes again. And when we quantified this, and I was skeptical that this was going to work, because what we're essentially doing is we're taking little bits of living human brain and we're dumping dead human brain on them, and I thought that would probably kill the slices. And we might see a lot of inflammation and the slices might die. But it was quite interesting because what we saw was we have to compare everything to media. So this is from the same people. So it's the same exact bit of tissue because some of these people were elderly. They could have had tau pathology. So if we look at people, the proportion of synapses that have oligomeric tau in the media condition where we're not exposing the slices to tau, it's very low. If we expose them to tau negative, so the immunodepleted homogenate, that stays low. But if we expose these slices to the tau containing homogenate, we start to see a significant increase in the proportion of postsynapses that have tau. So this is, each case is connected with the same color. So you can see that in all the cases, the amount of tau in postsynapses goes up if you treat with tau containing homogenate. This was also true with AT8, which is the phospho-tau, but you can see it's just much, much less. So the oligomeric tau seems to be the stuff that can really get into the postsynapses again. So I thought this was remarkable, that human living synapses can take up tau. What was even more remarkable was when we looked at the astrocytes. One of the reasons we look at astrocytes is because we're interested in the synapse-glia interactions, like I mentioned. And the other is that this GFAP antibody that we use is so amazing that we can stain anything with it, and it always works. So if we have an extra channel, we just say, let's see what the astrocytes are doing. So in this, we're looking at whether the astrocytes co-localize. So for example, here in the middle, you can see a tau. So sorry, you can see PSD95 inside a GFAP positive astrocyte, and you can see the same on the far right. And what was remarkable is we are always blind when we're doing these experiments. And so I love to be on the confocal, and I was doing some of Rob's imaging for him because he's also a clinician. He was back treating puppies, he's a vet. And so I was doing some of the imaging and I was blinded. And I said to him at the end of the day, look, some of those slices had a ton of astrocyte reactivity. It's probably just because we put dead brain on there. I can't get excited, but we'll see what happens. It turns out it was very specific. So the astrocyte reactivity was significantly increased only when that homogenate contained tau. So it wasn't just that the dead brain homogenate had cytokines or something in it, because when we removed the tau, it was the same as media. So the human tau-containing homogenate increased astrogliosis and increased that phagocytosis of synapses by astrocytes. So we thought that was very, very interesting. Now I'm going to move on to some unpublished data. I mentioned BIN1 as one of the risk factors. So of course, we've seen now, I've shown you in Alzheimer's and PSP, we think tau is spreading through synapses. But we, of course, want to know how that's happening. What could be mediating this so that we can maybe target that as therapeutic? And one of our first targets was BIN1, bridging integrator 1, which is one of the gene variants that's associated with late onset AD. In model systems, it's been shown to influence tau release and tau spread. There are some conflicting data, but we thought it was still, with the human genetics being so prominent, we thought it was worth looking in the human disease. So first we started with one of the other reasons we looked at this was not just other people's data, but we have a little bit of our data. These are our fly guys, Dr. James Catterson and Dr. Edmund Morpho, who do a ton of Drosophila work for us. And Drosophila, I don't know if any of you work with them, but they're beautiful for screening. So they grow really fast. We turn on human tau protein when they're in the adult stage, so we're avoiding any issues of development. And what we see this is tau off condition, and what we're doing here is we're looking at their activity. So this is the proportion of daytime sleep that's measured in these Drosophila activity monitors. And when the tau is off, they don't sleep too much during the day. When we turn the tau on, you get this huge phenotype in female flies of excessive daytime sleep. And then we ran a screen with a bunch of things we thought might influence this phenotype. And 2 of the top 3 were expressing BIN1 as well as tau, made things worse in the flies. And I've got, I hope, a little video here of the data. So this is the actual data. This is the proportion asleep is 0, awake is 0 and sleep is 1. And you could see the controls when the light phase comes on, they wake up and they wander around. But if you look at the tau flies, they're pretty sleepy, and the tau and BIN1 condition flies are even more sleepy during the day. So we have this exacerbation of a sleep phenotype in flies with BIN1. But of course, we then want to go back to the human condition because flies are a very artificial model system. So what Elizabeth's been doing lately is trying to understand whether tau and BIN1 are together in synapses where it might be influencing this spread. So she's again taken human postmortem Alzheimer's tissue, and she's looking at tau, synapses, and BIN1 protein. And what she's finding is that, of course, we still see tau in synapses. That's a very replicable result in our lab. BIN1 is present at about the same rate in our Alzheimer's and control cases. And there's a significant but small increase in synapses that contain both tau and BIN1 in Alzheimer's disease. But it wasn't really, the question was not just, are they in the same synapses, but could they be actually binding each other? So Elizabeth used a super resolution technique called FRET to start to get a handle on this, and Martí Colón-Cadena got this running in our lab. So FRET is based on a fluorescence principle called Forster resonance energy transfer, and it's using this idea that fluorophore spectra overlap. So what Elizabeth does is she labels either tau or BIN1 with a donor fluorophore that has an emission wavelength that overlaps substantially with the excitation wavelength of this fluorophore that she's stuck onto the other protein. And what happens is if you shine the light only to excite the donor protein, fluorophore, when you, for example, this is now a red fluorophore. So if you shine a 579 nanometer laser at a red fluorophore, it will glow red. That's what you expect, right? If you shine a 594 laser or a 579 laser at a far red fluorophore, nothing happens. But if this far red fluorophore happens to be within 10 nanometers of a red fluorophore whose spectra overlaps, then you can shine the red laser at it and you get far red light out. So you get the emission of the acceptor. But that only happens if the proteins are within 10 nanometers of each other. So with this metric, what Elizabeth was able to show is that when you look at synapses where you have tau and you have BIN1, within the presynaptic terminal, you get this signal. So the FRET signal is the signal when you're shining the light at only the donor fluorophore and you're recording only the acceptor fluorophore. So you get this signal, meaning that they're close enough to transfer that energy non-radiatively. And the proportion of synapses that have that FRET signal is very similar to the proportion that she counted that have both proteins. So this is some data suggesting that tau and BIN1 are close enough to each other to be actually interacting within individual human synapses. So this is a target now that we're taking back to model systems to see if we can manipulate and prevent this tau spread by manipulating BIN1. Now, in the last few minutes, I'm going to change, because I want to leave time for questions. So in the last few minutes, I'm going to change tack to synucleinopathies. These are some data from many years ago now, from when Martí Colón-Cadena visited during his PhD from Alberto Leo's lab, where we looked in dementia with Lewy bodies. We didn't have very many cases at the time, but the question was very similar to the question we asked about tau, which is, can alpha-synuclein potentially be spreading through the brain through transsynaptic spread? So Martí looked at synaptophysin, so presynapses, postsynapses, and phosphorylated alpha-synuclein. And what he looked for was whether synaptic pairs could contain alpha-synuclein. And what he found is that of the synapses that contain alpha-synuclein, very similar to the tau story, the majority of them contain it in the presynaptic terminal only, followed by pairs that contain alpha-synuclein, followed by postsynaptic alpha-synuclein only. So more recently, we decided, well, that's very interesting. We have some cases, and our case numbers have been growing for these synucleinopathies. And what we thought we'd do is see, since we know that there are co-pathologies and that alpha-synuclein can interact with tau in many different model systems, and there's been data showing them together in lesions in the brain using standard histology, we thought we'd just ask whether they might be interacting in the synapse. So Elizabeth, again, has done a small project where she looked at presynapses, phosphoalpha-synuclein, oligomeric tau, and whether they might be able to generate that fret signal, that sort of interaction signal within individual synapses. And we have a series of a few control cases that are matched to a few PDs and DLB cases. And she's looking in the putamen here because we knew there should be some pathology. And what she sees is that, as Marti observed, there are some synapses that contain alpha-synuclein. There are some synapses that contain oligomeric tau. There's no difference between the disease statuses here. And there are some synapses that she can find both of these pathological proteins together. Again, there's not really a significant change between the different diseases or the controls. But what she does find is that we get this fret signal. So when these two proteins are together, they generate a fret signal, meaning that alpha-synuclein and tau oligomers are close enough inside human individual presynaptic terminals to generate a fret signal, which I think is pretty cool. So if they're together, they do seem to be interacting. We don't really know what that means, whether this is part of any spreading phenomenon or part of any toxicity phenomenon, but we thought it was interesting and worth exploring further. So we're collaborating with Austin Millerwood up in McGill, who does some iPSC work, and we've sent them some synaptic alpha-synuclein and tau to play with in model systems and see what it looks like. So that's the data. I'm gonna wrap up for a couple of minutes so that we have time for questions. I hope I've convinced you that pathological tau is definitely present in synapses. I think we've shown that many, many times. It's very convincing. We've gone all the way down to EM level, and we're convinced. We are sure that tau spreads through the brain, at least in part through synaptic connections in model systems, and we have mostly indirect, but still, I think, convincing evidence that this could be happening in human disease. So I think at least one of the ways tau is moving through the brain is by jumping through synapses. There are no doubt other ways, probably involving glia, neurons dying, etc. But I think this is one way that it's happening. And at least in Alzheimer's disease, we have a little bit of new data suggesting that this tau spread and maybe toxicity could be something to do with BIN1, which is one of these risk genes. It's early days, unpublished, but we're excited to follow this up because that's sort of where we're going, trying to understand the mechanisms of this spread. And this is sort of a model of synapse pathogenesis. This is a figure that we made for Alzheimer's disease, but you could sort of replicate, put alpha-synuclein in there, and it almost is the same if you're thinking about synucleinopathies. So we know that synapse degeneration is really important for cognitive decline. We know that there's something to do with the glial changes in terms of disease risk in Alzheimer's disease, and we know that the inflammation occurs across the board in neuro and neurodegenerative diseases. So we think synapse-glia interactions are probably important. But the glia are not just bad, they're not just eating synapses, they might be pruning sick synapses, and they're certainly eating pathology, at least at various stages of the disease. So there seems to be a very complex relationship and something that I think is going to be difficult to therapeutically target with glia because they seem to be good and bad and at different disease stages and different brain regions. But I am convinced that if we could stop the spread of pathology, we should be able to slow or stop disease progression based on at least the animal data. You never know until it works in people, but I'm pretty convinced that that's a good path to take. So what we want is to help people ultimately. You've seen from all this that I'm just a complete nerd. I'm a neuroscientist. We sit in the lab with a microscope. But at the end of the day, I would love for what we do to work towards helping people. What we have now across all of these degenerative diseases is that your function, whether it's cognitive, motor, or both, starts off normal and then gets worse over time. And there's, for most of these diseases, not a lot we can do about that. There have been, since the early 1990s, at least for Alzheimer's disease and for Parkinson's, I can't remember exactly when, symptomatic treatments. So you can boost synaptic function, either with, in the case of Alzheimer's, cholinergic drugs, or you can give dopamine in the synucleinopathies when you have dopamine neuron loss. And that helps a bit, right? Your symptoms feel a bit better, but it doesn't stop this inexorable decline. So we call those symptomatic treatments. And that's really all we've had for decades and decades. What we've just gotten out in the Alzheimer's field, and I'm not taking any credit for this, just telling you, is there are disease-modifying treatments now that if we remove the amyloid plaques, you can slow the cognitive decline by about 30%. And that's certainly better than nothing. That's a disease-modifying treatment, but it's still not exactly what we want. We'd much rather have a way to stop the decline Or even better, we would want to not have that decline in the first place. So what I'm thinking, based on our data and lots of things in the field, is that stopping the spread of tau is probably a really good way of stopping that decline, because wherever the tau goes, the neurons die and the synapses die. And the synapse loss is really closely associated with your cognitive symptoms. Ultimately, I hope in the future, maybe a decade, maybe two, we'll get to this stage where we can profile people's genetics and their lifestyle risk factors, and we can know what pathology is coming for them, and we can give them a treatment so early that they never have a symptom. That's the dream. So we can all hope for that. And then, of course, even better, could we have a cure? People ask me this all the time. Are we going to have a cure for dementia? That's the hardest, right? Can we go from someone who already has lost, by the time of symptom onset, probably half the neurons in the important brain regions like entorhinal cortex? Can we do something that's so good that we boost and the circuits, maybe make new synapses? Can we actually make people better? That would be even better. I think that's going to be much harder to achieve. I have a lot of hope for the prevention, though. I think that's going to be achievable in the coming decades. So this is the final conclusions. And I wanted to just say thank you all for letting me blather on late at night here. So if it's not been that clear, I apologize. These are the shameless plugs. So if you're interested, if you have colleagues, friends, or family who have experience with dementia and want to know more about the neuroscience, I gave an entirely embarrassing, but I think at least accurate TEDx talk not long ago. And I've got a book that's coming out this year that's aimed at more sort of students and maybe nurses, people with lived experience who are keen on the neuroscience, but don't have the neuroscience background. So please do recommend that if you know anybody who'd be interested. So this is the thank you to all the people in the lab who are absolutely amazing. We're actually hiring in Edinburgh. We're looking for an institute director for our school, which is a big position. So if anybody's interested in that, please also get in touch with me. It's a big job, but we need somebody good. So please think about it. And thank you. I'm happy to take any questions. Maybe I could stop sharing, maybe.
Awesome, thank you. Questions? Anyone?
Hi, my name is Jeff Murphy. I'm a neuroscientist here in Michigan, and my group works a bit on Alzheimer's disease. So I really enjoyed your talk. Thinking about the astrocytes, do they die from the tau as well, or is it not toxic, or do we not know?
So we haven't counted the neurons. So in general, you get more reactive astrocytes in PSP and Alzheimer's disease. So they said they tend to, if anything, you grow more of them, or at least they become much more reactive and bigger, if nothing else. We tend to measure it with burden. The one time that we've done the direct experiment about whether tau is toxic would have been in the slice cultures. And we didn't count the astrocytes, but we did see them growing, or at least the amount of space in the brain taken up by them went up. So I don't think that the tau is probably killing the astrocytes, or if it is, it's killing some of them, but more are reacting because they can breed, they can multiply. So no, I don't know if it's toxic to the astrocytes specifically. In culture, sure, you can give a lot of tau and it will kill cells. But in the brain, I think they're able to just divide and make up for it.
Yeah. And then you had said that the oligomeric form is probably the form that's the most toxic. Is that, do you think, because it's the one that can travel?
Probably. So I think that we used to think that tangles were toxic because they're ugly and they're definitive lesions, and we thought they must be killing cells. And there are things called ghost tangles. So you can see in the brain where a neuron had a tangle and it died. So the tangle's left, but the neuron's not. So clearly some neurons with tangles die. But I guess ultimately we found out in a sort of serendipitous way. We went to test that hypothesis specifically in mice, where we could watch over time in the living brain. We could watch the tangles form and we could watch the cells and see if they died. But it turns out that cells that got the tangles were the ones that survived and not the ones that didn't. So we all went looking, but in the end, it was a surprise, and that's always fun. And it's probably my favorite surprise of my career so far. That was when I was still working with Brad and Alex Calignon. But then it kind of makes sense in retrospect, because the tau fibrils themselves are like bomb proof. You have to use really stringent detergents to break up those fibrils. So they're almost inert, they're so hard and heavy. Whereas the oligomers and the sort of soluble forms can, as you say, they can move, they can float around, they can interact with receptors, whereas the fibrils are just so solid that I don't think they can do too much directly. They seem to be able to seed new aggregates, which is not great. but they don't seem to be able to themselves interact with too much.
That's cool. Thank you.
Mhm. So I have a quick question. If is there any neuron type specificity in terms of when tau is more likely to spread from the pre to the postsynaptic? So from excitatory to inhibitory or inhibitory to inhibitory, or does it need spines or is it just bin one expression? Like, are there rules for any of that spread?
Yeah, it's a great question. It's not one that we have directly addressed in our human tissue, but there's been a lot of great work in animal models. And it's and just sort of from the standard histology as well, how pathology in Alzheimer's is more common in excitatory neurons. And in mice, it spreads more through excitatory neurons. And in fact, the more excitation, the more excited a neuron is, the more tau it spits out. So excitation itself, or sort of neuronal activity itself, also is involved in tau spread, we think. So it's not to say that that inhibitory neurons aren't affected. There are some data looking at various types of inhibitory interneurons, and they do have tau pathology. One of the reasons we focus on excitatory neurons is because we're looking at this distal spread from one brain region to another, and it's usually the principal excitatory neurons in the cortex that are the ones that send the axons to other brain regions. So from that perspective, I think for tau spread, we definitely are focusing in on those excitatory circuits. I think locally the inhibitory circuits probably spread tau amongst themselves as well, but we haven't looked really in detail at that in the human tissue.
So I mean, many mouse models can show pretty large intrinsic changes in inhibitory neurons. So I guess those could be coming about because they're losing some of the excitatory inputs onto them or many other triggering factors that may not have to do with direct tau spread. Is that fair?
Yeah, yeah, probably, but I'm sure there are, and I just don't follow the data that closely. I have looked at inhibitory synapses in Alzheimer's around plaques, but not in the context of
Mhm.
tau, and we do see changes, we do see loss. So there, I think the neurons are
Yeah.
definitely affected, and that change in excitation inhibition balance. There's a lot of work going on in that and
Mhm.
sort of epilepsy relationships to Alzheimer's, and we have colleagues in Edinburgh like Sam Booker who are really interested in
Mhm.
inhibitory neurons, like really interested, and they're looking at them in the context of Alzheimer's pathology. So I'm sure they're playing a role. I just don't think it's probably a huge role in that transsynaptic, transregional spread, unless it's indirect, like through modulating the activity.
Got it. Mike?
Hey, very nice talk. I was curious about a lot of it is really seems selective for the T22 antibody, and it's not something we've used very much. So I was just curious how that compares to other antibody staining. Are these really mostly synaptic, or are they still seeing the nerve fibrillary tangles? And would that suggest that they're recognizing conformation that's early misfolding but before phosphorylation if they're not being detected well by AT8 or other phosphoantibodies?
Yeah, such a good question. I don't have a super satisfying answer. So the short answer is T22 does see other things. It's not just synaptic. And I'm sure that we did look at like the proportion of T22 signal that was in synapses versus other things, but it's also, we can see it in tangle looking structures. We can see it in neuropil threads. We can see it in dystrophic neurites around plaques. The sort of bad news is that the T22 we use came directly from Rockets. When we buy the commercial one, we can't get it to work on our fixed tissue. I think it's our tissue goes through that whole baking process and everything. So we've had no luck with TOC1 or the other sort of oligomer antibodies that we've tried. So we have to beg, and Rockets is very kind and sends us that. The good news is though, we see this across lots of different tau antibodies. So pretty much any pathological tau antibody that we try, we can find it at synapses, some proportion or other. So T22 is the most predominant in that one study we did head to head, but some of it could have been the secondaries and things like that. So, and we definitely see AT8 reproducibly in synapses as well as in neurons. We've seen p-tau217 recently, which is a very hot topic because it's one that gets out into the blood. So it's a biomarker, and we see that in both dystrophic neurites and synapses, and we're doing a whole study on that. So I think if you're interested in synaptic tau, you can kind of look at a total tau antibody and you can see it in the synapses as well. So kind of anything you use will probably show up, and it's a little bit messy as to what proportion is in which form.
I see. I was just curious about how they relate to neuritic plaques, because neuritic plaques really seem quite related to cognitive decline, and they accumulate a ton of tau. And are there synapses near there? Are they kind of more positive than synapses further away from? Like, I'm just wondering if those are a particular site of tau release or internalization.
That's such a great question, Mike, and that's something I was writing a grant about this morning. So the plaques themselves, whether they're neuritic or not, are always in mice and every human I've ever looked at. If it has a dense core, there's synapse loss around it. So dense core plaques cause proximal synapse loss, and they have synaptic accumulation of oligomeric amyloid beta in the proximity of plaques. We've only once looked at tau in synapses with respect to plaques, and we didn't see much, but it was a long time ago, and it was a total tau antibody, and it wasn't the main point of the study. So we're trying to do that more thoroughly now, because I think you're right. You get right around those plaques, you get those dystrophic neurites, and they're probably leaky, and they get a lot of tau in them, and there's synapse loss, so that might be a spot of tau spread as well. We're actively trying to figure that out.
Cool. Thank you.
So maybe you want to review my grant. I've got to send in a couple of months.
That sounds exciting.
Tara, there's a question in the chat. Is there any laminar specificity to the spread of tau?
Yes. So if you think about the entorhinal cortex, which is where the tau pathology starts very early, it's the layer 2 neurons. In fact, real and positive layer 2 neurons are really susceptible to tau pathology and early death. So in EC, there's very much laminar specificity. And the other brain regions, I don't know the data as well. We see tau pathology through all the cortical layers, but I don't know if one arrives earlier than the other. For example, the Braak staging is pretty crude. It's like, is there a single tangle in that brain region? If so, that's a positive region. It doesn't really get at laminar specificity. So I'm not aware of, I would say just anecdotally that layer 5 neurons and layer 2, 3 pyramidal, the big ones, tend to be the most common, but I'm not aware of any systematic other specificity of dot spread.
Great. Other questions? Anyone? If not, just thank you again, Tara. That was wonderful, and thank you for taking the time, and I hope to see you again soon.
Thanks, y'all. Sorry for talking fast. I always talk too fast.
No, that was brilliant. No, appreciate it.
Take care.
Thank you.